therapy. So what I want to talk to you about was the work we've been doing over many years actually in the lab, trying to use mouse models to, first of all, study ccRCC in general and molecular mechanisms of ccRCC, but I'd like to focus today actually on developing therapies for clear cell renal cell carcinoma. Do you actually see my mouse with the cursor?
Not right now, but eventually might.
No.
No, okay. Seems like it doesn't have a pointer function. Wait, just bear with me 1 second. Do you see the laser pointer?
No, we can see it.
Yep, super good. Makes my life a bit easier. Okay, so yeah, I think all of you, I can skip the introduction to clear cell renal cell carcinoma, a frequent tumor, and obviously VHL inactivation is one of the key molecular features of the disease. VHL is truncal, and as you all know, one of the best described and probably one of the most important oncogenic functions of loss of VHL is the activation of the hypoxia-inducible transcription factors, and HIF1 and HIF2 get together, and these induce a bunch of gene transcription profiles, many of which are obviously important for various aspects of the development of the tumors. And I'll touch a little bit on some of the work we've done on looking at the roles of the HIFs in immune crosstalk and trying to use this to improve checkpoint therapy. So I think I also don't need to introduce any of you to this concept that checkpoint therapies have really revolutionized the treatment of metastatic clear cell renal cell carcinoma in recent years. And of course, it's now at the stage where combination therapies are the way to go, either in the form of anti-PD-1 or anti-PD-L1 together with tyrosine kinase inhibitors, or in the combination of checkpoint inhibition with PD-1, CTLA-4. And these have both advantages and disadvantages, and that is that the combination TKI therapies generally seem to induce more frequent partial responses, but more rarely complete responses in ccRCC patients, whereas the combined immune checkpoint therapies show actually lower overall initial responses, but tend to improve the chances of longer term survival. Of course, this is said very quickly, and there's exceptions to these rules, of course, but I think what is very positive is that the curve's always moving upwards and to the right, that patients are surviving longer and longer, but the downside of this is that these therapies are still clearly not optimal for all patients, and there are many patients who still go on to succumb to their disease. So there's a lot of work still to be done, and I'd like to tell you a little bit about some of the mouse model work we've done as a model of resistance to checkpoint therapy. Of course, the other hot topic in ccRCC is the great work that's been done over many years from Peloton and collaborators, Bill Kalin and Jim Bugarolis, in preclinical settings, showing that the HIF2 inhibitor works very nicely in some preclinical kidney cancer models, but also preempting that it's likely that not all tumors will respond equally well or at all even to HIF2 inhibitors. And this, I think, has been borne out in the first clinical trials that are going on and are still going on, that there are some patients that you see here that are responding very, very nicely over a long period of time to these drugs, but other patients are either not responding so well or not responding for so long. So HIF2 inhibitors certainly is a new armory in the fight against kidney cancer, but I think that's still not going to be the whole story. So coming to our own work and how we're trying to contribute to this field, it comes back to the generation of a mouse model of clear cell renal cell carcinoma that we described about 5 years ago now, which is based on the renal epithelial specific inducible deletion of VHL P53 and RB. And this is a somewhat unusual combination for ccRCC genetics, but it very broadly seems to reflect the complicated pattern of gains and losses or chromosomal gains and losses of genes that regulate cell cycle progression. And these mice over time develop clear cell renal cell carcinomas that really look like ccRCCs in humans. They have this intricate vasculature that you see with CD31 and von Willebrand factor stainings, for example. And importantly, they show many molecular overlaps with the human disease. So this is an RNA-seq data on the left and proteomic data on the right, the Venn diagram showing the overlap between the genes that are differentially regulated in the mouse tumors compared to normal kidney, overlapping with the human tumors versus the normal kidney. And in both cases, there's actually quite a remarkable overlap. So we think it's histologically and molecularly quite a good model of clear cell renal cell carcinoma, albeit a model, and may not reflect all types. One of the first things we did historically was wondered whether, well, if it's a model of ccRCC, we'd like it to behave therapeutically like human ccRCC, and that was what we tested in this experiment that we described, where we tested at the time what was sort of the first line and state-of-the-art therapy, being sunitinib followed by everolimus, the mTOR inhibitor. And kind of out of interest, we chose a drug that had been described by Greg Semenza's lab, acroflavin, as being an inhibitor of HIF1 and HIF2, and had shown some nice responses in mouse models. And what we observed was actually that, like in humans, there's kind of a heterogeneity of the responses of tumors in these models, that some of them, for example, were responding, or not responding in this case, sorry, to sunitinib, whereas these tumors in these mice were kept quite nicely in check with sunitinib. And then we saw tumors that would respond to everolimus, but not to acroflavin, and vice versa. Even within the same animal, there were tumors that did or didn't respond to everolimus or to acroflavin. And so that led us to this sort of traffic light view of the independent tumors in each mouse, that they were really behaving differently in terms of regression or growth to these different drugs. So it kind of encouraged us that, well, at least this model seems to mimic some of the aspects of therapeutic responses that are seen in human ccRCC, so maybe we can use it to investigate new drugs. And before I tell you about that, I'd just like to diverge back on the topic of the HIF inhibitors. At the time we were doing this, the HIF2 inhibitors sort of came on the scene, and it became very clear from human genetics that HIF2 seemed to be the more oncogenic force, and HIF1 rather tumor suppressive, if anything. So we did a genetic experiment where we then deleted HIF1 and HIF2 in the background of this tumor model, and again induced gene deletion and waited to see what happened. And what we saw was that in VPR, this is VHL-P53, this is the shorthand we use. This is the same curve you saw before. If we introduced HIF1 deletion, we completely abolished tumor formation, or reduce the effect of VHL, let's say, abolish the effect of VHL on promoting tumor formation, whereas HIF2 deletion kind of had an intermediate effect, both on the incidence as well as on the number of tumors we saw per mouse, suggesting that in this model, I think very clearly genetically, that HIF1 is really the oncogenic force and less so HIF2. And this was sort of borne out in therapeutic studies that we subsequently did, where each line here represents the growth of an individual tumor in multiple different mice, either untreated. These tumors basically always grow exponentially, treated either with acroflavin as first-line therapy, now treated with the HIF2 inhibitor, PT2399 in this case, or with an inhibitor of sphingosine signaling, which had also been reported to inhibit HIFs. And what we saw was, again, in keeping with the genetic data, that the HIF2 therapy, if we just look at this is now looking at change in tumor volume, so basically the growth rate during the therapy period, you see that HIF2 inhibitor therapy had absolutely nothing, or had no effect on tumor growth, whereas the drug that theoretically inhibits both HIF1 and HIF2 did have a therapeutic effect, and fingolimod actually in many cases showed even nice tumor regressions, albeit temporary ones. So I think that sort of suggests, at least in this model, that the bad guy, the oncogenic force, is HIF1, both genetically and in pharmacology, and not HIF2. One thing we did notice in RNA sequencing was that we...
Ian, can I ask a question about that?
Yeah, sure.
So have you tried to take the PR mice and then cross them with the LOX HIF1 DPA to see if that is sufficient to get RCC?
Yeah, that's a good question. Whether overexpressing HIF1 in the P53RB background. No, we haven't done that. Yeah, that's sort of the reverse experiment. I suspect it. Yeah, I don't know. I suspect it won't, because I think VHL.
Yeah, so that's so. I think Billy Kim had done some work in the past, but I think he had done the experiments largely in the liver tissue, not so much in the kidney tissue, where he had tried to model what VHL loss does compared to HIF1 alone or HIF2 alone, and then a combination of both. And from what I remember, I think he needed both to model the disease. So maybe I'm just thinking in terms of the genetic makeup of the tumor, right? So this might be a very, if you compare to the human genetics, it might be representative of very advanced RCC tumor, perhaps. And so in that context, if the biological circuit is flips in some which way.
Yeah.
I'm just trying to reconcile with the clinical data, basically.
Yeah, you're absolutely right. I mean, one important limitation of the model, of course, is that we delete HIF1 and HIF2 right at the very start of tumor formation. And so I think it probably what it tells us is that you need HIF1 in the early stages because it's pro-angiogenic, it changes metabolism, it does a lot of things. But what we don't know what would happen is if we would delete HIF1 later after tumors already form, and if we do the same with HIF2. So it may be that early on it's necessary for tumor formation and then it's lost. And the same, by the same argument, HIF2 may be the opposite. So yeah, I think it doesn't, our data doesn't speak against HIF2 being important later. But clearly HIF1 is important early. I think we can conclude that. Okay, so one thing we noticed in the RNA sequencing actually was that in gene set enrichment analysis, we kept getting all of these signatures popping up, particularly strongly in the tumor model where HIF2 was deleted, and these had to do with immune activation signatures. There were lots of different ones. Here's one with T-cell activation, and you can sort of appreciate it's more yellow under the VPRH2 than the VPR tumors, and that led us to look more closely at the immune cells, and indeed what we could see, albeit fairly subtly, was that there was generally a bit more, or a statistically significant increase at least, in the number of CD8 and or CD8 T cells in the tumors, but more strikingly in the level of activation markers of things like perforin or CD69. So suggesting that losing HIF2 would actually promote anti-tumor activity, and that may be one of the reasons why there was actually a delay in tumor onset. A caveat of that is that if we think about this pharmacologically, of course, if you want to inhibit HIF2, then doing that with a drug like the HIF2 inhibitors, belzutifan, for example, will also inhibit HIF2 in all of the other immune cells. And it's become very clear in recent years that HIF2 is really necessary for efficient T cell activation. And I think our therapeutic studies sort of bear this out. When we did the same analysis looking at the number of T cells in the HIF2 treated mice, we actually saw that there were fewer T cells here than in grey in the untreated. And in particular, markers again of activation, CD69 and perforin, were reduced upon HIF2 therapy. So I think that's one flag that will be very interesting to see what happens in the human clinical trials of checkpoint inhibitors plus HIF2. And my feeling is that HIF2 therapy may not be such a good idea in that case, because we may actually be suppressing the T cell activation that is trying to be activated by the checkpoint therapy. So speaking of checkpoint therapy, that's exactly what we decided to do, of course, with the mouse model. And just to show you, these are very long-term experiments that were done very patiently, but mostly by us and in later times a little bit by Xin helping. The mouse model is quite tricky to work with. You have to feed the mice for 2 weeks at 6 weeks of age, and then basically mice will develop tumors sort of stochastically over the next sometime between 5 and 12, 14 months. These have to be followed by ultrasound on a mouse-by-mouse basis to see when each animal gets a tumor. Then we switch to MRI imaging to get better therapeutic data once we start doing the therapy. So these mice, then we tried to learn as much as we could from them, harvesting all of the organs, sampling various things for flow cytometry, for histology. We made cell lines out of the tumors whenever we could, and I'll show you some of that in a minute, as well as cryopreservation of splenocytes, and I'll show you why that was important. So we first thought, well, what we do is we test sort of, in our mind, the most promising therapy at the time, which was PD-1 CTLA-4 therapy, and protocols were established for the mouse. We looked in our RNA sequencing and we saw, for example, here in red that the tumors tended to have more PD-1 and CTLA-4 expression, and at least upregulation of expression of the ligands of CTLA-4. So we thought we'd try this experiment. And basically what happened was absolutely nothing. So the mice more or less ignored the drug. Here you see the growth curves of the combined checkpoint therapy to mimic ipilimumab and nivolumab. And again, once you crunch the numbers, there was no difference in tumor growth rates. When we looked more closely at the tumor microenvironment through flow cytometry of the tumors, we were quite surprised actually to see that, if anything, there were fewer tumors compared to the control or the untreated mice, fewer T cells. And this was borne out in both CD4 and CD8. And surprisingly, actually, there were a tendency to have more myeloid cells. So essentially we were getting the opposite of what we would have liked or what we might have hoped to have seen. We thought we might be able to promote T-cell infiltration into these relatively T-cell cold tumors, but in fact what we did was actually promote myeloid infiltration, which tends to be more immunosuppressive. So what Asin then did was tested a whole bunch of other therapies that we thought might act to tip the balance of the tumor immune microenvironment and promote T-cell activation and infiltration. So she tried the aquaflavin that I mentioned before, the HIF-1, HIF-2 inhibitor, the HIF-2 inhibitor, which is sort of similar to the belzutifan pembrolizumab clinical trial that's going on. We tried actually to immunodeplete putative myeloid-derived suppressor cells, and basically this didn't work, so you can ignore this therapy. We tried cabozantinib, which is in line with the COSMIC 313 trial. We were trying like immunogenic chemotherapy using cisplatin as well as STING activators and inhibitors for various reasons that I won't go into just now. And to cut a long story short, again, nothing really happened here. So we tested all of these drugs. The only thing that really saw that jumped out was, apart from a few regressions in aquaflavin, was that cabozantinib slowed down the tumor growth. But I should say here that this therapeutic combination was very toxic. And we lost, I think, 3 out of the 5 mice, 3 out of the 6 mice that were dosed with this regime. And I think this growth inhibition is probably more to do with the TKI activity rather than a combination with checkpoint therapy. And again, you see this here, that the only significant decrease was actually with cabozantinib. So I can go very quickly through all the flow cytometry data and just tell you that we looked again in all of these mice at all of the same markers we looked at before, and essentially nothing really happened. We didn't see any rescue of CD4 or CD8 T-cell infiltration, unfortunately and surprisingly. And again, there was no real change of this generally increased expression or the increased abundance of myeloid cells. So that was somewhat disappointing. Of course, what we were worried about was that whether there was even recognition of the tumor cells at all by the T cells, given these findings. So Asin devised an experiment where she tried to basically take away the contribution of the immune microenvironment and just expose T cells from the same mouse to tumor cells from the same mouse. So she took advantage of this resource that she generated of frozen splenocytes and clear cell renal cell carcinoma lines from basically a matched set of resources from individual mice that were either treated or not with therapy. She then took the T cell or the splenocytes into culture and activated T cells or not with CD3, CD28 beads, and then cultured the T cells alone or together with the ccRCC cell lines from the same animal. And then we did flow cytometry for a whole bunch of different markers of CD4 and CD8 T cell activation and exhaustion. And what you see is sort of exemplified in these pictures. If we focus just for simplicity on the CD8, looking here at interferon, we have a control mouse example on the left and a mouse from one of the large groups of immune checkpoint inhibitor therapy treated mice. And you see that the staining of interferon gamma alone is quite low. When the T cells are cultured by themselves, when we put them together with tumor cells from control treated animals, we see no activation, but we see quite a robust activation when the T cells and the tumor cells were isolated from the mice that were treated with checkpoint therapy. And in many cases, we actually see a similar phenomenon, for example, here with granzyme B, where there's a stronger activation once the cell's already pre-activated with the CD3, CD28, and then co-cultured. So this was indicative of an activation, and I'm going to cut really a lot of data short and show it in this heat map, where basically the more red the squares are, the more activation we had of the T cells with that particular marker. So it's the ratio of the median fluorescence intensity of the co-culture versus the single culture. And looking at CD4 on the left, CD8 on the right, what you basically see, all these markers are much more strongly expressed or more abundantly expressed when the T cells from the immune therapy treated mice are mixed together with the tumor cells. And one exception here, that's actually anecdotal because we only have one mouse, is that the HIF-2 inhibitor actually didn't show any activation, which I think is consistent with the data that I showed you before. And again, makes me quite nervous about the human clinical trials. So obviously,
that's a quick question. So is it that the tumor cells and the splenocytes are all from untreated or all from the ICI treated, or is there any mixing and matching? Meaning, I guess my question is this an effect on the tumor cell side or on the immune cell side? That if you took a tumor cell, tumors from mice that were treated with ICI and, splenocytes from untreated, how would that?
Yeah, no, I understand where the question's coming from. We didn't do that because we can't do it because the mouse model is on a mixed genetic background. So that would just be an allogeneic setting, and there is certainly activation when you put a foreign, T cell with a foreign tumor cell.
So it has to be autologous, yeah.
It has to be autologous in this setting. So that's the way we're sort of limited in doing this. And we're also limited in the number of tumors from which we successfully managed to get cell lines, which was dependent again on how big the tumor was and various other factors. So it's kind of all we had.
No, that makes sense.
But I think nonetheless sort of reasonably convincing that the checkpoint therapy is at least uncovering an immune response, even if, so it's a systemic immune response because the T cells were in the spleen, even if we're not seeing a strong immune response within the tumors. So that actually brings me to one very quick conclusion. That is just to remind you that the mouse model is actually variably sensitive to sunitinib and everolimus, just like in human setting. We discovered it's sensitive to fingolimod. It's resistant to HIF2 inhibitor and very strongly resistant to combined checkpoint therapy, at least therapeutically. So it's kind of then a glass half full or half empty situation where the model can either be seen as being bad because it's not really mimicking common therapies of ccRCC. It's not responding well to checkpoint therapy, but we actually tend to think of it more as a good model of therapy-resistant ccRCC. And so the other conclusion is that the combination of kind of our in vivo and ex vivo studies really tend to implicate the tumor microenvironment as suppressing this adaptive immunity that is somehow being uncovered by PD-1 CTLA-4 checkpoint therapy. So that's what we've been focusing on in this respect, is to try and better understand the tumor microenvironment, and we leaned very heavily here on many of the studies that some of you know, you David in particular, looking at single cell sequencing of human ccRCC. And so we did the same thing basically in the mouse. Shown in the top left here is the experiment where we took we did single cell RNA-seq from 3 tumors and 1 normal sample. Normally in blue, what you already see in the UMAP plots is that there's sort of an imbalance on the left and the right side in terms of the amount of blue to pink ratio. And when we went and looked at the different identities of these different clusters, we realized that on the right, which was very enriched in the tumors, was actually the myeloid population, and the lymphocyte populations were here on the left. When you sort of crunch through the numbers and look at the relative distributions of the immune system in the normal versus the tumor setting, we very clearly see this sort of shift that lymphocytes dominate in the normal setting with relatively few myeloid cells, but this is completely reversed, and actually the lymphocytes are in the minority, and the majority of the cells in the tumor microenvironment are myeloid, with particularly striking increases of this population of neutrophils here in red, and another population that I'll mention a little bit in a second, these Ly6C2 high monocyte macrophage cells. This sort of appears to be a sort of consistent with a classical immunosuppressive microenvironment, in that the CD8 to Treg ratio is very low in the tumors, and the CD8 to myeloid ratio is also true. So approximately for every single CD8 positive T cell we have about 12 myeloid cells, or myeloid-derived lineage cells in the tumor. On the T cell side, basically I can summarize this very quickly in these violin plots. There's very little to say, actually. So there's a very weak signature of exhausted T cells, but a very, very non-existent signature of T cell activation. So here we have CD8 activation markers, here CD4 activation markers, for example, granzyme and perforin, IL-2, interferon. Blue is, again, the normal, and pink are the tumors. And you basically see that there's really no T cell activation on the CD8 or CD4 side. And there's sort of a hint of an exhausted phenotype with transcription factors that mediate exhaustion, like TOX and EMES. There's a few cells in there, but and PD, basically very, very few cells that are really expressing these strong exhaustion markers, either on the CD4 or CD8 side. And I think that's maybe consistent with all of the stainings we've done. What's a bit more interesting is on the myeloid side, as I said, these are the cells that are enriched. And when looking through very quickly a broad range of potentially immunosuppressive markers, we see that the ligands for PD-1 and CTLA-4 are relatively poorly expressed or weakly expressed in the different myeloid cells. But what's particularly strong is things like LGALS3 that you see here in the violin plots. that lots of different populations, including actually the ccRCC cells themselves, are expressing galectin-3, which is interesting because LAG3 is one of the checkpoint molecules that's of interest therapeutically. We also see strong expression of things like TGF-beta and IL-1 beta, less extent TNF, which are all known immunosuppressive signaling molecules and cytokines in the immune cell microenvironment. You see, for example, the different populations of the myeloid cells are expressing different amounts of neutrophils. What's interesting, as I alluded to a minute ago, is this yellow population of these Ly6C2 high population of cells. This is the Ly6C2 itself, expression itself is high in the tumors. ITGAM is actually CD11B, and the classic sort of marker combination in mouse for myeloid-derived suppressor cells is high expression of CD11B and high expression of Ly6C2. So at least at the RNA-seq level, this sort of looks a bit similar, like an putative MDSC phenotype. There's also strong expression of markers like fibronectin-1 and F13A1, some of the markers that have been reported to be high in macrophages, tumor-associated macrophages that correlate with poor patient outcome and aggressive responses or aggressive behaviors of human tumors. So this population seems to be a bit of a mix of aberrantly expressed markers that really shouldn't be there, and in fact are very absent in the normal setting. So we think this is a misdifferentiation of myeloid cells down an MDSC lineage into kind of a TAM2-like phenotype. So these are all consistent like RNA signatures, I should say, of potential myeloid-mediated immune suppression. We looked then immunohistochemically at different markers of myeloid cells and indeed see them at very early stages of tumor formation, so in very, very small lesions. So we think that there's probably a recruitment of these cells to the tumor. So we're interested in following that up more and did RNA sequencing actually of tumor cell lines compared to primary mouse renal epithelial cells to look for differential expression. We see things we like to see, like MYC and E2F, that are probably driven by HIF, amongst other things, by RB deletion in the model, hypoxia and glycolysis by VHL deletion. So this sort of checks out. What was interesting was that we saw interferon gamma and interferon alpha signatures. And here you see in the bottom the individual genes in these signatures, red being tumor cell lines and normal renal epithelium in black. And all of these genes, basically strong interferon signature, are upregulated, including some very interesting cytokines at the end here, CXCL10, CCL5, CXCL2, and MIF. These are all known to recruit macrophages, recruit actually myeloid cells in general, I should say. So we followed that up, taking conditioned medium from these cells and actually measuring these cytokines. You can detect them quite nicely in the secreted medium from the supernatant from these cells. And given that many of them are predicted to mediate macrophage migration, we actually tested that by inducing bone marrow-derived macrophages and mixing them together with the conditioned medium in a transwell assay, and then basically measuring the number of macrophages that cross the transwell and are chemo-attracted to the conditioned medium. And here you see, compared to a positive control for this migration, that actually the tumor cell supernatants do very well, and they very strongly and potently recruit macrophages. What's more than that is that they appear to, the conditioned medium appears to mediate this misdifferentiation that I was talking about. So in this experiment, basically we mixed conditioned medium with bone marrow cells and let them differentiate over time and just see what happens in culture. And what happens on the myeloid-derived suppressor cell side is that we see that compared to a positive control, that again, the conditioned medium induces MDSC-like phenotype. Ly6C high, CD11B high, like the RNA sequencing, the single-cell RNA-seq signature I showed you, and at the same time induces the proliferation and the differentiation of macrophages, both of the TAM1-like phenotype, CD206 negative, or TAM2-like phenotype, CD206 positive. So this is suggestive of a potentially immunosuppressive phenotype of these cells in terms of T cell activation. So we then did the following experiment where we basically took the differentiated bone marrow cells and made, again, more conditioned medium out of the same conditioned medium, so it's sort of doubly conditioned, as well as the myeloid cells themselves, and then cultured them with activated CD3, CD28, activated CD8 T cells from the spleen. Now this is from just a black 6 mouse in this case, to look at generally T cell suppression. These experiments are trace them with crystal violet, so the non-activated T cells basically don't proliferate. The T cells only proliferate. You see beautifully the dye dilution in the FACS plots here, indicative proliferation. And then when we add in more and more T cells, the ratio of bone marrow cells to T cells, what we see is that basically there's a successive suppression of the T cell proliferation, and this is quantifiable as the differentiation index. So basically the degree of inhibition. So the stronger the degree of inhibition here, the higher the number, and basically the more of these derived bone marrow cells we have in the ratio to T cells, the more inhibition there is. And as I said before, the bone marrow cells greatly outweigh the tumor cells, the T cells, sorry, in the tumor microenvironment, and already at a ratio of 1:1, we have a very, very strong inhibition. So I think there's good evidence that there's a crosstalk between the tumor cells, differentiated bone marrow cells, and the suppression of T cells. This also works with human cells. So here are 3 different human ccRCC cell lines that are commonly used. And again, these induce the differentiation of MMDSCs and induce macrophage proliferation and somewhat differentiation to the TAM2-like phenotype. So that's something we're following up. What we think, and I cut this story very short, actually, for the interest of time, because I want to talk about something else in the last part, is that we've identified a number of different molecules that we're very interested in that may mediate this crosstalk between the different cells of the normal monocyte lineage, causing differentiation to this sort of MMDSC and TAM2-like phenotype. And these include CCL5, we've got CXCL2, CXCL10 may actually directly try and activate T cells, and MIF is a very interesting candidate which signals through different receptors, and they've all been implicated in playing a strong role in regulating the function of these myeloid cells, or the aberrant function of myeloid cells in tumor microenvironments. A strong candidate is galectin-3 that I showed you is expressed by many of these different cell types in the TME, as well as many of these secreted cytokines. So we've got a lot of candidates now that we're following up on exactly these cell culture assays, as well as in future mouse models that we can test to see whether we can try and remove the immunosuppressive aspect of the myeloid microenvironment and improve T cell therapies. So that was what I want to say about that particular aspect of the model. I don't know if there are any questions before we move on to the last part of the talk. That doesn't seem to be the case. That's good. So what I'd like to tell you about in the last 15 minutes or so, maybe less, is the work that we've been doing recently that we will hopefully be accepted for publication in the coming days. And that is to try and convince you that DNA damage shouldn't be forgotten about as a potential therapeutic target in ccRCC. So what's very well clinically established, and all of you will know this, is that ccRCC is one of the tumors that responds very poorly or not at all to chemotherapies. And this has always been a little bit of a mystery why that would be the case. And I think it's particularly a mystery when you look at the genetics of ccRCC. So this is the mutational frequency of different genes in primary and metastatic tumors. VHL, PBRM1, SETD2, BAP1, KDM5C, and P53 are kind of the most frequently mutated genes. And all of these play important roles in DNA damage repair or DNA damage repair signaling. So VHL mutation in replication stress and a bunch of other functions. And basically all of these sort of classic epigenetic regulators all also regulate different aspects of DNA damage repair and signaling, as I said. And as well as that, metastatic ccRCC sequencing studies have shown that sort of really classic DNA damage repair molecules are also mutated in about 20% of the cases. So I think there's sort of a genetic predisposition, if you like, that there's something going on with DNA damage in clear cell renal cell carcinoma. And we actually came across this through independent means. This is actually the same RNA sequencing experiment I showed you earlier, where we took the mouse VPR ccRCC cell lines and compared them to primary mouse renal epithelial cells. And we also took human ccRCC cell lines, just standard cell lines that we all know and use, and compared them to primary mouse renal proximal tubule epithelial cells. So we did RNA-seq, we did gene set enrichment analyses, and then looked at overlapping terms. And this is this upsetter plot is basically a multi-dimensional Venn diagram where the overlapping gene, so in this case they're not genes, they're overlapping reactome gene set enrichment pathways between these different comparisons are shown here. So at the far end, basically there's not so much that doesn't overlap between comparisons of mouse versus normal and the individual human cell lines versus the normal. But in this left-hand side of the graph, we find all of these different terms that are commonly upregulated in the tumor cells to normal in all of the different conditions. And when we looked at these, we were struck that basically all of them had something to do with DNA damage. It's basically every sort of reactome term you can imagine that has to do with DNA damage repair and other processes. And particularly of relevance to what I'm going to tell you is this term of activation of ATR in response to replication stress. So this suggested at least that there's something going on with the DNA damage response and a strong signature in the transcriptome of ccRCC. Just to show you how this looks in the sort of classic gene set enrichment plots, we've chosen here the term DNA repair. You see the enrichment in the mouse setting, in the human setting compared to normal. And because it was cell culture, we worried that it may be an artifact of cell culture, but indeed, when we look in RNA-seq of tumor biopsies versus normal biopsies in the mouse setting, we see the same enrichment. And also in the TCGA kidney cancer data set, we see that when we take the matched normals. So that was an RNA signature. We wanted to, of course, check if there's really DNA damage, and we used immunostaining for gamma H2X, a common marker. And we see very clearly in the mouse model that most of the tumors have a lot of damage, although this is quite a scatter of the extent of number of cells strongly expressing gamma H2X. And similarly in biopsies of human patients where we could have matched normal and ccRCC, we always saw that there was kind of a strong gamma H2X. So I think DNA damage is clearly going on in ccRCC. And this caught our attention because of the...
Can I ask a follow-up on that aspect? If you were to do the same analysis for, given all the extent of aneuploidy and replication stress in general in all cancers, if you were to rank where kidney ranks among others, would you see that the DNA repair or DNA damage response is significantly higher in RCC, or exceptionally different from other cancers?
No, we did that. Actually, that was one of the things we did for the revisions of our paper. We did the same bioinformatic analysis of about, I think, 24 different TCGA sets of tumor and normal. And what's interesting there is ccRCC doesn't really stand out. So it's neither stronger nor weaker in general, I think, very quickly said. So I think, like other tumors, DNA damage is a feature of ccRCC. That's probably the conclusion.
Thank you.
So I think what it does tell us is that the chemotherapy doesn't work. The absence of a chemotherapy response is not because ccRCC doesn't really have a DNA damage issue. It does, but somehow the therapy is not working. But given everything I just said just now as well, we were interested in new classes of drugs that actually have been designed to target exactly these sort of problems that tumor cells have with excessive DNA damage. And these are small molecule inhibitors of the molecules that signal DNA damage, namely ATR, ATM, and DNA PK. And this we did together with Merck. And here I have a little disclosure that the following research that will soon hopefully be published was actually conducted in the context of a research collaboration with Merck here in Germany. It was a really fantastic exploratory collaboration. We actually started off studying HIF2, and this merged into a DNA damage collaboration through various reasons. Anyway, what we did was we tested basically the sensitivity of ccRCC cell lines, either VHL null in the control setting or VHL rescue, to a bunch of different drugs, including 2 ATR inhibitors on the left, ATM, DNA PK, as well as a series of conventional chemotherapeutic agents. And what we found was, somewhat surprisingly, given the prior literature, that the presence or absence of VHL made no difference, but the cells could quite happily be killed in culture by chemotherapy. And in particular, we saw this very strong sensitivity of the ccRCC cells to inhibitors of ATR, and that's what we followed up on in this whole story. The mechanism is a long and complicated story, but to cut it short, we basically showed, as one might expect, that ATR induces replication stress in ccRCC cells, so ATR inhibitor here, this M4344, which is an orally available drug. You see in these pulse labeling DNA fiber assays where the drug was given throughout the entire pulse, that basically what you can do is measure the length of the different pulses, and this tells you different features of DNA repair. Basically, the reduction in the average track length of labeled replicating DNA is always indicative of replication stress, also the ratio between the two different pulses. So basically what we then went on to show was, and I won't show you the data because as I said, it's very complicated, that what tends to happen in ccRCC cell lines is that they kind of ignore the replication stress depending on the dose, and they progress through into G2M. This causes problems with cell cycle progression, but cells often continue to divide. That causes micronuclei, as well as DNA damage in G1. So over time, the replication stress and the inhibition of the sensing of replication stress causes many different DNA damage abnormalities in ccRCC cells. So we then tested that as monotherapy in ccRCC xenografts, and we were happy to see that in two different models, 7860 and A498, that we got therapeutic responses just with monotherapy. So this was very encouraging. But we wanted to look and see whether we could find any more therapeutic synergies based on what many people have done in many different cancers. So we basically we went back to the start and tested all of these different drugs that are inhibitors of ATR, ATM, and DNA PK. And we chose sensitizing doses that caused a sort of a sublethal inhibition of cell proliferation of these drugs, and then combined them in synergy assays where we did dose response curves with a whole range of different chemotherapeutic agents. And what jumped out of all of this in these synergy assays was basically cisplatin and carboplatin, as well as some other drugs like decitabine and olaparib. And just to show you in the follow-up here, you see this sort of classic dose concentration curves where at varying concentrations of the ATR inhibitor, we see the classic left shift in cisplatin sensitivity and vice versa. And basically, when you crunch all of these numbers together, you can calculate through an isobologram the coefficient of drug inhibition. And essentially anything that falls under the diagonal here is indicative of synergy, and you can put a number on this, and the closer it is to 0 than 1, the more synergy. So we did these same assays for the combinations with cisplatin, carboplatin, and olaparib with 5 different ccRCC cell lines, and they all show a very nice therapeutic synergy. So we went on and tested this, now choosing ATR plus cisplatin because it's quite advanced in clinical trials. We tested this in the xenograft setting and now shifted the dosing rather than daily dosing of ATR. This was basically a weekly dosing where we give a dose of cisplatin followed the next morning by ATR. And what we could see very nicely, the red is the combined therapy, both in 7860 and ATR, that we got a very strong synergy, or actually induced a therapeutic response in the case of A498. So I think this is quite a nice finding, because what we're doing here is basically taking models that are chemotherapy resistant, so that both cell lines both xenografts are more or less resistant to cisplatin, and making them now sensitive when we add ATR. So I think there's maybe a lesson here for the situation in human ccRCC, where we know that the tumours are generally resistant to chemotherapy, that we may be able to sensitize them by adding in an ATR inhibitor. We then tested this combination, and again, we only tested the double combination, as I said, because these are very long and involved experiments. We just wanted to see if it would work. We tested in the autochthonous mouse model and saw different things, basically. So, as I said, the untreated mice the tumours always grow exponentially over time. Some mice basically ignored the chemotherapy, the combined therapy, whereas others showed really beautiful and strong shrinkages. And you see this in the plots here, the growth over time, again, exponential growth in control, and a range of different responses in the mouse tumor model, which I think fits also with what we saw and what I showed you at the start, that the different drugs and different therapies are causing different effects. So we don't really know why this is. As I told you, we know that the tumours in the mouse model have a large range of the degree of gamma H2X positivity. And so we thought we'd take a look at that. And I sort of naively thought that the more gamma H2X positive, the more likely it was that the tumour would respond to this damaging chemotherapy. And it turns out the opposite is the case, even though we only have a few mice here. The tumours that continued to grow all had very high DNA damage, whereas those that shrank had low DNA damage. And we think that maybe one thing could be that these tumours are sort of used to living with a lot of DNA damage throughout their evolution. They had DNA damage, so they just learned to ignore it and weren't really that fussed when they got more DNA damage from ATR and cisplatin. The other thing that was very interesting was this idea of using chemotherapy to induce immune responses by causing DNA damage, which signals to the innate immune an adaptive immune response. And in short, we did the flow cytometry of all of these tumours and basically saw nothing other than a macrophage infiltration in many of the tumours, which I think is probably consistent with induction of cell death and necrosis. So that was something that was a good idea, but didn't work out. So to conclude for this part and the whole talk, so I think what we've shown is that endogenous DNA damage is really a feature of ccRCC, and we should think about ATR as a novel class of molecular target. I think it gives us the chance to think about more smart sort of combination chemotherapies that may be useful in certain cases where patients have failed all other available therapies. This is really an active field of clinical research, so there's currently 49 ongoing or recently finished phase 1 and phase 2 clinical trials using ATR inhibitors. The ATR inhibitor alone is very, very well tolerated. In combination with some other agents, there are emerging toxicities, but this is something that has to be figured out. There's the thought to think about whether there's underlying genetics or molecular features that influence the sensitivity of tumours. And as I said, this issue with any chemotherapy, of course, is always efficacy versus tolerability that would have to be studied in clinical trials in many different settings. But I think ccRCC is one that we should consider moving forward and typically hasn't been considered. So on that note, I would like to stop and thank you all for your attention, and thank actually many of the people in the lab who are shown here on a recent lab hike who did a lot of this work, as well as many of the sponsors and many great local and international collaborators. So on that note, thank you very much, and I will sign off and hand over to any questions. I'm not sure how to escape from. There we go.
Maybe I can lead off while people are gathering thoughts. One, I probably have already tried this, and maybe I already asked you this in the past. Have you? I know that the direct transplant of the cell lines from the mice, probably they don't engraft in immune proficient recipients again. But have you thought about any sustained passaging in culture, or even like any random mutagenesis type approaches to see if you can create a cell line out of those that acquires naturally some epigenetic or genetic alteration that will now allow them to engraft more rapidly and make life a little bit easier rather than the 12 to 18-month latency with tumors that we're seeing right now?
Yeah, we haven't done that, and we haven't done it for a reason, and that is that the cell lines, what we have done, the ccRCC, the mouse ccRCC cell lines, when we've taken them in culture and then put them back into subcutaneous growth as xenografts in SCID mice, the histology is very sarcomatoid. They look like fibroblasts, like tumors, and we don't know what that means actually. You could either argue, well, that's a very aggressive sort of form of ccRCC, and histologically that's really bad news, but it doesn't look like the original tumors. Whereas, 7860 cells still look like ccRCC, A498 still looks like ccRCC, but these look much more like tumors we've made in the past where we transform fibroblasts.
What about a secondary orthotopic transplant into the kidney?
Haven't done that. That we haven't done. Yeah. No, we haven't thought of that actually. No. I just got a little bit nervous once we saw these cell lines, and I didn't know.
I think it might be a good, just you don't probably have to enroll 20 mice or whatever, but for a quick 2 or 3 animals, if you test that, I think it might not be a bad idea, because I think there's a clear difference in engraftment rate in the orthotopic kidney versus subcutaneous. So subcutaneous tumors, I think, tend to require a lot more vascular support and probably just have a completely different oxygen sensing profile, etc. The other thing is, I think, going by what Jim was able to successfully achieve with the intracardiac injection. So that would be another alternative approach to just see if you can model advanced RCC form by doing a cardiac injection. So,
yeah,
so they might just engraft at better rates using those.
haven't done that. I actually don't really know that work of Jim. Is that published?
There's a Cell Reports paper that they published with the entire biorepository of like.
It's the Roy Elias paper.
Yeah. Okay. Yeah. Yeah. Okay. Yeah, we haven't tried those sort of experiments. We tried to we tried to avoid further models where we don't have control of the setting. And I think the beauty of the autochthonous model is really that the immune system is really co-evolving, but it's painful, as you say. It'd be nice to have a faster system.
Sure.
Yeah.
Yes, I thought from the first sort of half, the PT2399 plus checkpoint blockade and sort of aberration of T cell effects is like pretty concerning, and like maybe, so I know it's one mouse, but like kind of a good word of caution. Do you see that with the acroflavin as well? Like, was it just specifically HIF2 inhibition, or did you see that with sort of pan-HIF inhibition or HIF1 alpha inhibition as well?
That's a good point. We did, so in the first study we did where we didn't have checkpoint therapy, we did also see a suppression with acroflavin, which is, the problem with acroflavin is it's a terrible drug. It does a lot of things.
Yeah. And
it has horrible pharmacological properties. It's been shown to work in models and things, but it's actually a DNA damaging agent. So I'm kind of nervous about overinterpreting anything with acroflavin now. So yes, we saw an inhibition. I don't know if that's related to T cells don't like having a DNA intercalating agent stuck into them. Yeah, I think that the human studies are going to, as you say, I'm nervous, but I'm going to be interested to see whether there's a competing activity. Maybe it's okay if you change the scheduling, that you're not inhibiting T cells at the same time as you're trying to activate them. I'm not sure. I don't know if there's preliminary data yet. I talked to a few people, but
yeah, we have a little bit on the human side as well, which again, and Anna Goldrath had these papers where certainly it seemed like they didn't discriminate between HIF1 or HIF2, but that was sort of advantageous to have HIF activity in infiltrating T cells. And there's a separate sort of component as well, which how much that regulates ERV expression, which might be an immune target. But I think there's probably multiple kind of floating around points of evidence that, and this seems like in line with that. But
I think Randy Johnson has done some really nice studies actually with mouse models and all sorts of engineered T cells and CAR T cells. And I think it's quite convincing that inhibiting HIF is not a good idea if you want to promote immunity. I think, or T cell mediated immunity, and not to mention what it does as well in the myeloid cells. And basically, immune cells really need HIFs, I think.
What are your thoughts on like the radiation? Because there's like ATR inhibitors that are used in concert with radiation that are being studied right now, and how like this is perceived as like a radio-resistant tumor, even in the context of having this much DNA damage, I guess. Yeah,
no, I think it's a very good point. It's the one stimulus that we didn't test, but basically everything else we tested where we couldn't reliably induce sort of DNA double strand breaks, we were seeing, well, cisplatin's a bit different actually, but we were seeing good synergy. I think it's certainly worth testing. It's, as you say, there are studies using ATR as a radiosensitizer, ATR inhibitors as a radiosensitizer, and I think if that would work, it's certainly very attractive, because one of the problems, of course, with chemotherapy is if you add a drug that further sensitizes the chemotherapy, it certainly will have more side effects. And that's coming out of the early clinical trials with cisplatin and things like that the patients are getting dose-limiting side effects in the trials. But radio for isolated metastases plus ATR inhibitor, I think, is an attractive idea. But we don't have any preclinical data to support that there is a sensitization in ccRCC. It's something we should do. Yeah. Is anyone doing any ATR inhibitor studies at your institutions, or is that off the radar?
We are, yeah.
For RCC specifically, Ritesh?
Not for RCC, but yeah, like we had the ATR radiation trial, and I think I enrolled an RCC patient on it because it was sort of a pan cancer, just getting radiation.
Yeah.
In a sense.
How does it look actually in that setting? pan-cancer.
I mean, it's interesting because you only get drug for like the 4 days. You only get drug during the radiation period, actually. So again, I don't know what the data looks like, but from a tolerability standpoint, it's like 3 or 4 days of drug while you're getting something done.
And I think in general the drugs are.
And if it potentiates, then it's that really becomes like an interesting thought process of like specifically where you're radiating, right? Bone versus somewhere else. But yeah, I mean, it's really compelling to kind of look at this kind of thing. Yeah.
I wonder. Yeah, I mean, the problem we have is I don't know how you deliver stereotactic radiation to a mouse. I don't know if anyone has a mouse facility that can do that. I'd like to. It'd be nice to do it in the tumors, actually.
Well, you can imagine also, I mean, just thinking like longer term, right? Like, for example, in prostate, where there's lutetium that's approved as far as some type of emitter, and whether that could be sort of combined in the context of this. I mean, we have a open trial right now of Nivo plus lutetium labeled anti-CA9 that's paired also with some type of imaging. But you can imagine, again, if you have some type of emitter that's there with adding this in, maybe that would help just by thinking about it.
Yeah, sure, that's interesting. Yeah, something more targeted. The second stimulus needs to be more targeted than a general chemotherapy that destroys every other tissue in your body. Yeah.
It'll be hard, I think, to sometimes go back to cisplatin, I guess, in combination with something like this, just because, in our patient population, cisplatin is nephrotoxic. So the ability to just give, even if we developed a study of like cisplatin plus ATR in this study, it'd be, I think, very hard to administer in RCC patients, just thinking about it.
Yeah.
I mean, maybe just like de novo metastatic disease where they still have 2 kidneys.
Yeah, right. Yeah.
Yeah.
Just from like a thought perspective, right? How to do a study.
Yeah, sure. No, I completely agree with you. So that, we also identified, we identified carboplatin as another hit. We identified decitabine, which is actually quite interesting and unexpected, and we identified olaparib. And we followed up in the 7860 model and did the olaparib plus ATR therapy. No, not olaparib, was another PARP inhibitor that Merck had, and nothing happened. So very, very clear synergy in cell culture, but no response in the model. And decitabine, interestingly, we see a very strong response of decitabine by itself, but no synergy, despite very, very clear synergy in cell culture. And that we really followed that up actually with complicated cell biology. And decitabine's a little bit tricky because of the dosing, particularly in mice. It's really hard to keep a pharmacological dose. I think in humans it needs to be delivered IV as well, if I'm not mistaken. So yeah, exactly for that reason, we're also not really keen on giving cisplatin and ATR inhibitor to patients. Yeah.
I have to jump off. I apologize for another meeting, but thank you. That was
great. Thank you so much.
You're welcome. My pleasure. Thanks for listening to the discussion, and